Okay, so as we talked about in class, chapter 19 is about how genes are turned on and off. So to start off, we're going to do a quick little review about chromatin and DNA and how DNA is packaged. So this is a review. So chromatin, the DNA protein complex found in, and that is eukaryotes. So we talked about this when we talked about cell division, that is DNA loosely wrapped around proteins. So that's what this picture is showing you here. So here's your DNA molecule. Here's your DNA wrapped around your histone proteins. All right. So then that is chromatin. How do we get from chromatin to a chromosome? Remember I said that they coil and tightly pack together to form a short, stocky little chromosome. So this picture is showing you how they do that. So the DNA is wrapped around the histone. One of these little beads, which is the DNA wrapped around the histone, is called a nucleosome. So it's one of those little beads. And then those nucleosomes coil and pack closer together. And then so this right here is this here. And so then that coils and loops. And then those coils coil and loop, and then eventually form a chromosome. So you've got very tightly packed DNA wrapped around proteins in your, just notice here it says metaphase chromosome. All right, so you can look at it backwards too. This section is this section right here. This section is this section right here. All right, and so on eventually until you get your DNA molecule. So that's how DNA is packaged. So let's turn the page. So DNA within cells undergoes a variety of changes as it proceeds through the cell cycle. So going from chromatin to condensing and forming chromosomes and so on in the cell cycle. We call them prophase. It's highly diffuse, but as the cell prepares to divide, it becomes highly condensed, forming those chromosomes. So proteins called, what are the proteins that the DNA wraps around called? Histones are responsible for the first level of DNA packing. The mass of histone is nearly equal to the mass of DNA. So there's a lot of histones, a lot of DNA. So DNA, how does it bind to histones to wrap around it? So DNA is negatively charged. And histones contain a proportion of positively charged blank. Well, histones are proteins. What are proteins made of? What are they made of? Amino acids. And enable easy binding of the histones to the DNA. Histones play a very important role in organizing the DNA, and they're good at it. Thus, it's a likely reason why histone genes, the genes that code for making histones, have been conserved throughout the generations in the course of evolution because it's very helpful to keep the DNA organized. The structure of histones are very similar among eukaryotes and between eukaryotes and prokaryotes as well. So a relatedness between all living things. So electron micrographs, remember those are pictures taken by electron microscope, show unfolded chromatin, and they look like beads on a string. These beads are referred to as, remember what they're called, going back over here, they are called nucleosomes. So that, which can be considered the basic unit of DNA packing. And the string would be, the string wrapped around is DNA. So then, looking at nucleosomes and DNA packing, a nucleosome is a piece of DNA wrapped around a protein core. All right, the protein core is the histone. This DNA histone association remains intact throughout the cell cycle. So even when it condenses into chromosomes, you still have the DNA wrapped around histones. When it uncondenses and kind of spreads out again back into chromatin, it still has DNA wrapped around histones. Histones only leave the DNA very briefly during, and that is what we talked about in chapter 16, and that is during DNA replication. With very few exceptions, histones stay with the DNA during DNA transcription. So with a few exceptions there. Nucleosome interaction and DNA packing. The next level of DNA packing takes place between the histone tails of one nucleosome and the nucleosomes to either side. The interactions between these cause the DNA to coil even tighter. So we're condensing. As they continue to coil and fold, eventually the DNA resembles that of the metaphase chromosome. Okay, so here's some new things. Heterochromatin versus euchromatin. So during interphase, some of the DNA remains condensed as you would normally see it in metaphase. So as the centrosomes, the telomeres, and other regions of the chromosome, this is called heterochromatin. So some chromatin stays condensed. This is called heterochromosome to distinguish it from euchromatin, which condenses and relaxes with the cell cycle. So euchromatin condenses and relaxes. This one remains condensed. All right, so that's a new thing. So let's flip over the page. So as you can imagine, heterochromatin is rarely transcribed, which means that you're not going to make a protein from it. So if it never unwinds and always stays tightly packed, then you really can't read it and make an mRNA and do protein synthesis. So heterochromatin, we don't really use it to make a protein. So then the structural organization of chromatin is important then in helping regulate gene expression. So this is what I said is the nitty-gritty of what Chapter 19 we're going to talk about is turning genes on and off and how that occurs. And one thing has to do with if it ever does heterochromatin that doesn't loosen back out and relax, that can't be expressed, and so therefore that affects gene expression. Also, the location of a gene's promoter relative to nucleosomes and to sites where DNA attaches to the chromosome scaffold or nuclear lamina can affect whether or not it is transcribed. We're going to build on that and talk about that as we go. So I'll talk about the chromosome scaffold and the nuclear lamina as we get into this. Research indicates that chemical modification to the histones and DNA of chromatin influence chromatin structure and gene expression. So my question to you is, do you remember we talked about a chemical modification and turning genes on and off in a specific example with genetics. Remember the mice, and it depended upon if you got the gene from mom or dad, whether or not you had it. And so remember when we added the methyl groups turn the gene off and doesn't allow it to be expressed. That would be an example of chemical modification. So let's look at this a little bit more in detail. Gene expression can be regulated at any stage, but the key step is transcription. So obviously, if you're not going to make a protein, then why even go through transcription? So it makes sense to regulate the first stage. All organisms must regulate which genes are expressed at a given time because it's very wasteful to make every gene all the time. A multicellular organism's cells undergo and this is differentiation, which is specialization in form and function. So they become specialized in terms of what they're going to do. So a skin cell versus a stomach cell versus an eye cell and a neuron. So therefore, we have differential gene expression. Every cell has different genes that are turned on and off at different times. So differences between cell types result from differential gene expression. Why a neuron is different than a muscle cell is what genes are turned on when. The expression of different genes by cells within the same genome. So what does that mean? All the cells of your body are from the same organism, but we have differential expression, and different genes are turned on and off in different cells of our body, and therefore we have different types of cells that can perform different functions for us. In each type of differentiated cell, a unique subset of genes is expressed. So we can put example, skin cell versus neuron. All right, there are two different types of cells. Different genes are expressed in those cells. Many key stages of gene expression can be regulated in eukaryotic cells. So how do we regulate this gene expression? So gene expression is really protein synthesis. What you're really doing is regulating protein synthesis that the proteins are going to give you the trait, all right, and you're going to get something's going to happen in the cell as a result of having that protein or that gene expressed. So let's look at these regulations here. What this picture is showing you, here's your chromatin. It says a signal. So we're going to see that signal molecules, like different molecules in cell signaling from chapter 11, can trigger a gene to be turned on and be transcribed. So let's just look at this. This is the nucleus. So this is a eukaryotic cell. So here's your chromatin. Looking at a piece of DNA here, this is the gene from right here to right here. All right. And so here's transcription. You make your RNA, and I'm going to fill in the blanks. My blue here are the introns that are going to be cut out. So the blue parts are cut out, and the exons, the coding part, are spliced together. So therefore, what can happen is in transcription, and even before transcription, in these boxes here, these boxes are ways in which we can regulate gene expression by allowing these things to happen or not happen. So every box along the way tells you what's happening. So we can do chromatin modification, where DNA unpacking involving histone acetylation and DNA methylation. We're going to talk about that here. So we can regulate whether or not we're going to get a protein at the end right at the beginning by regulating whether or not the chromatin is going to unravel, so it exposes the gene. If it never unravels and that gene stays wrapped around a histone, then that gene is never going to be expressed. So we'll talk about these two specific ways later, but that is thing one. Thing two is the gene being transcribed, so we can somehow regulate transcription or RNA processing. And then remember that the mRNA goes out after processing, the introns are cut out, you get your cap and a tail, goes out the nuclear pore, here's your mRNA and the cytoplasm. It goes to the ribosome and translation occurs, so we can regulate translation. Here's your polypeptide. Sometimes the polypeptide undergoes further modification to become the active protein, so we can prevent that from happening. And then eventually the active protein can be degraded so that it doesn't continually do what it's doing in the cell. So that allows us to say, okay, if we need to still do it, we need another signal to make another protein, and so on. And then remember that there are enzymes in the cytoplasm that degrade the mRNA. And so all of this are areas in which we can regulate whether or not your protein is going to be made in the first place. All right, so we're going to talk a little bit about that. So regulation of chromatin structure. Genes within highly packed with heterochromatin are not usually expressed because they never uncoil, and so therefore the gene is never exposed, so transcription cannot happen. So chemical modifications, it's not supposed to be chemical modifications to histones and DNA of chromatin influence chromatin structure and gene expression. So this is where we're going to talk about histone acetylation and DNA methylation here. So both of these have opposite effects. One is going to allow the gene to be exposed so that you can do transcription. One is not going to. So, so histone modification. In histone acetylation, so that was that first one, you add acetyl groups are attached to the positively charged lysines. Remember, lysines is a type of amino acid in histone tails. So the cell adds acetyl groups, all right, to the histone tails. The tails, by the way, let's go back to this picture. That's these guys here hanging off of the histone proteins, histone tails. They're part of the histone proteins. This process seems to loosen chromatin structure, thereby promoting the initiation of, what are we going to do? If it loosens it up, it exposes the gene, so what can we start? Transcription. All right. Let's turn the page over here. So that's what this picture here is showing you. Here's your histone tail here. So this is your histone molecule, DNA wrapped around it, and that completely is called, the whole thing is called a nucleosome, right? So here's your tails. And so what it says is that when it's packed, it says unacetylated histones, so they don't, no acetyl groups are attached. They are more tightly packed. Look at after acetyl groups, that's what these guys here, the acetyl groups are these little dots here. Acetyl groups equals little dots here. Loosely packed equals exposed genes. And if the genes are exposed, then you can do transcription. So this is a good thing to turn the gene on. So let's say the gene is right here to right here. Now it's exposed, and we can go through transcription and make the mRNA, then process it, then do translation and get our protein. So acetylation kind of turns the gene on. It helps in turning it on. The opposite of that is DNA methylation. So DNA methylation, the addition of methyl groups to certain bases in DNA, is associated with reduced transcription in some species like us. In some species, DNA methylation causes long-term inactivation of genes in cellular differentiation. So that means the specialization of the cells. The same genes in different tissues show that the genes are usually more heavily methylated in cells in which they are not expressed. So I was talking about digestive enzymes and how your eye cells have genes to make digestive enzymes, but they would always be turned off. And so they are heavily methylated. And so therefore, because they're never turned on, they're heavily methylated, and so therefore it never unravels and transcription never occurs, and you don't ever make those digestive proteins in your cells of your eyes. So we talked about this in genomic imprinting. I mentioned that at the beginning of the notes. Methylation turns off either the maternal or paternal allele of certain genes at the start of development. And so therefore, we've talked about that, adding methyl groups to that. All right, so DNA methylation turns it off. Acetylation of the histone proteins turns it on. So although the chromatin modifications just discussed do not alter DNA sequences, so we're not changing the DNA sequence, all these cells that never turn the gene on have the sequence there, so they can be passed on to future generations of cells. So the sequence is always passed on, and you can pass it on to your kids and so on. The inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence is called epigenetic inheritance. So this has to do with the genomic imprinting and the methylation. So, for instance, when we talked about whether or not you get it from your maternal or paternal alleles, so the methyl groups of one of the alleles that are added to that gene can be passed on to the next generation. So the next generation has the gene, but they don't exhibit it because of the methyl groups attached to it, and so that's called epigenetic inheritance. Regulation of transcription, and so far here we've talked about regulating whether or not the gene is going to be exposed. So next we can regulate transcription. Is the mRNA going to be made from the DNA now that the gene is exposed? So chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind to the transcription machinery. So remember the promoter region where the transcription factors and everything binds to get RNA polymerase to make your mRNA. So organization of a typical eukaryotic gene. So let's turn the page here. All right, associated with most eukaryotic genes are control elements, segments of non-coding DNA that help regulate transcription by binding certain proteins. All right, so control elements and the proteins they bind are critical to the precise regulation of gene expression in different cell types. So if we look at the gene, so here is our gene. All right, and here's the introns and exons, so this is the non-coding region and so on. This is the promoter region, here's the termination, and here's the poly A sequence at the end. So this is considered upstream from the gene, this is considered downstream, and so this is the gene right here. So let's just look at that first before we look at anything else, what we know. So then, during transcription, we make a copy and make an mRNA strand. But remember first that we take and copy also the introns. And so then we have to do RNA processing. So therefore, RNA processing takes out the introns, splices together the exons, and puts a cap and a tail on. So we have the cap and the tail. And here's your gene, and so it's showing you here, this is your finished mRNA after RNA processing, so transcription RNA processing. Here's your start codon, here's your stop codon, all right, and that's what's going to be read in the codons in the groups of 3 at the ribosome. All right, so now what I want to do is look at upstream, so here's your promoter region where everything binds. We have these things called proximal control elements. They're sections of the DNA. Proximal means close to the gene. Distal is farther away. All right, so then we have, these guys are upstream. Distal control elements are also called enhancers. All right, so that's what we're going to talk about, that these guys are instrumental, these control elements and the proteins that bind to them. We're going to see that there are certain proteins that bind to these, are critical to the precise regulation of gene expression in different cell types. All right, so we're going to look at that. So let's continue on in our notes, and we're going to come back and forth to this picture and look at some other pictures as well. So the roles of transcription factors. To initiate transcription, eukaryotic RNA polymerase requires the assistance of proteins called, remember, transcription factors. General transcription factors are essential for the transcription of all protein coding genes. Transcription factors bind to specific DNA sequences and or other regulatory proteins. In eukaryotes, high levels of transcription of particular genes depend upon control elements interacting with specific transcription factors. So let's build on that. So we know that transcription factors have to bind. They bind to the promoter region, and that helps RNA polymerase know where to bind and transcribe and make your mRNA. So we're going to add in this new piece of information. So proximal control elements, proximal, remember, are close to the gene, are located close to the promoter, which is at the beginning of the gene there. Distal control elements, groups of which are called enhancers, oops, that's an NH, may be far away from a gene or even an intron. Some transcription factors are activators. An activator is a protein that binds to an enhancer and stimulates transcription of a gene. So you have to have these molecules called enhancers or activators that bind to one of these enhancers and that helps to stimulate the transcription. So basically, different cells have different activators. And so therefore, if you don't have, the cell doesn't have an activator to bind to one of these control elements, then the gene doesn't get turned on. If there is that appropriate activator, it can bind and turn the gene on. So that's the big picture here. So that's what the next picture shows. So let's look at this picture here that shows these activators. And so this is kind of like the same part. Here's your gene with the introns and exons, and here are your enhancer, the distal control elements where activators can bind. So that's what we're looking at here. So now we can make it in different colors here. So these are the activators right here. So these molecules right here are activators. And then you have where they're binding. What do they call it? Make them blue on the other page. Here are the control elements. And this whole thing is called the enhancer, made out of these different control elements where the activators can bind. All right, so I'm going to put here these, whoops, I should probably write in the same color with activators. The activators bind to the enhancer. And the enhancer, let's get the right color here. The enhancer we can write as part of the DNA. So down over here is your promoter region. And remember, this is where that has the TATA box and where the transcription factors bind so that RNA polymerase can bind. So in order for that all to happen, you have to have activators. And so therefore, if we look from here to here, what's happening is this right here, this is still your promoter region right here, all right, and the TATA box right here. All right, and this is your gene, so that part's right. And so this part here folds over so that the enhancer regions right here are folded up on top kind of the promoter region. And remember, the enhancer has the activators attached to it. So I'm going to put here's your activators. Same colors here, so it's binding, bending over there. All right, so now let's look at these new things. So we have this guy here. Let's make him pink. So this is DNA bending protein. So it helps the DNA to bend, all right? And the purpose of that is it brings the bound activators closer to the promoter. So you can see here that they're right on top of each other. All right, and so in comes these mediator proteins and transcription factors. So let's see here, let me get purple, I'll use, what color am I used? In red. All right, so these guys right here, group of mediator proteins, these guys interact, let's say, with proteins at the promoter. And these guys are the transcription factors that we learned about with transcription that bind to the TATA box, but we're going to see how that all works in just a second. All right, and so we're going to look at more details here. All right, so that's what we have going on. So these guys are coming in. Now look at here. What happens is the transcription factors and the activators bind. So here we have, this is the transcription factor, was up here in purple, right? Let's get all of our things here. So I'm going to put these guys in red. All right, so the mediator proteins. And then let's review here the enhancer region right here. And then you still have the activators. Now notice here what's going on is the activators in orange are binding to the transcription factors and the mediator proteins. And so what's going to happen is when we said that to the TATA box, remember, which is right here, it's kind of underneath here, RNA polymerase can bind once this has happened. So you get the transcription factors and these mediator proteins, these all of these together trigger the RNA polymerase to bind and start transcription. And so therefore, let's write a few things about the activators here. So activators were orange here. So let's write that the activators bind to transcription factors and mediator proteins forming, what does it form? This little guy here, a transcription initiation complex. So we're initiating the process of transcription. And so this is how the RNA polymerase, I'll put that in yellow, this is how the RNA polymerase 2 knows where to bind and transcription can happen. So now, how does this relate to turning on and off genes? So remember, that's what we were talking about, is how does this control transcription? Well, this process and the binding of the transcription factors will not occur unless you have the right activators. So if the cell doesn't have the right activators, the activators will never bind, the transcription factors will never bind, and RNA polymerase will never bind. All right, so that's how it controls. Now there are activators, but there are also repressors that will molecules that can bind here that prevent this from happening. So they're called repressors. So some transcription factors function as repressors, inhibiting the expression of a particular gene. And some activators and repressors act indirectly by influencing the chromatin structure as well, and the structure of, remember, if the chromatin is still all bound up and condensed, it can't unwind, and therefore transcription cannot happen. So let's look at an example of this in a cell. So a particular combination of control elements can activate transcription only when the appropriate activators are present. And so that's what I was just saying here. If these guys aren't present, then this can't happen and transcription doesn't happen. So two examples here. So let's just look at this. So here's your control element, and the control element is a place where activators bind, remember, and collectively they're called enhancer regions. Now also I want to write here the combination is important in regulating gene transcription. All right, so what do I mean by combination? So what we're looking at is two pieces of DNA. one that makes albumin, and codes for making the protein albumin, and one that's crystalline. Albumin is made in the liver, and crystalline is made in the lens of your eye. And so, so what I mean by the combination, when we look at these control elements, the different shades are supposed to be different colors. So you could have this one yellow as one kind of control element. I'll leave, let's see, I'll do one in green. Or no, let's not do green because I already used green. Let's do purple. This one purple, and this one red. So what does that mean? It binds different activators can bind to that. So the colors mean that different activators bind to kind of quote unquote turn on the albumin gene. And the crystalline gene is turned on when the appropriate activators bond to their control elements, which are different. So, so that was supposed to be yellow. This is orange. It's kind of hard to see. That's yellow and this is orange. Then there's still the purple. That one's the same. So they have a similar control element. And then a different one. Let's do this one pink. Okay, so what it means is the combination. The combination is important because these guys need three different activators. These guys need three different activators. One is in common, but they need different activators for the yellow versus orange or the red versus pink to quote unquote turn on these genes. All right, so let's write another thing about the control elements is these only the appropriate activators can bind. So only appropriate activators can bind. So what does that mean? I'm doing colors. That's the purpose of my colors. All right. So, so albumin. Let's look at this. I'm gonna put down for these two genes. Let's put down here that both liver and lens cells have genes for making albumin. And crystalline. So all cells in the body have genes for making these guys here. They don't necessarily need to be made, these proteins, in every single cell of the body. So how is it that the albumin gene is turned on in the liver and the crystalline gene is turned off? And the lens cell turns on the crystalline gene, but has the albumin gene turned off. And so what we're looking at, here's your liver cell nucleus and your lens cell. So let's look at what's available here. So notice floating around here, you have different, these guys are activators. So that what this means available activators. Let's do, where's my pen? So these guys are available activators floating around. And so let's color code them as they correspond to these colors here. So this one, let's see, this one is going to be red. So those are red. All right. We also have gray, or sorry, purple. These guys here, I made the gray purple here. All right, so we have purple. All right, so remember, this is the albumin gene. So the albumin gene has right here the purple. All right, in the center, it has the yellow, Remember, it's folded over, so if I were to undo this, it would be yellow, purple, and then this is red. And notice here, what happens is the red binds to the red control element, the purple can bind to the purple, and then notice here that the yellow, this is a transcription factor, so an actual transcription factor binds to that. It's not an activator. And so what happens is you have three things that bind to these control elements, which means that it can turn on itself. The mediator proteins and transcription factors bind, RNA polymerase binds, and the albumin gene is expressed. The crystalline gene is not expressed because you don't have the right combination of activators. So if the crystalline gene is orange, purple, pink, orange, purple, and pink, you don't have enough activators. So it's showing you here that there's purple, but they also have the red. They don't have an orange one or a pink one, and so this one is not expressed because there's, let's put it here, not the appropriate activators. So therefore, in your liver cell, then you don't make the crystalline gene because your liver cell doesn't make the appropriate activators. All right. So then when we look at the lens cell, the available activators here are here. All right. And that's all these guys here. These guys, let's write here that these are different activators than in the liver cell. So let's color code them. So there's pink. These guys are supposed to be pink. They have the purple. Purple. Let's do this one purple. I think that's the right shape. And then they also have orange. Okay, so remember that the albumin gene had the yellow, the purple, and the pink control elements. They only have the purple out of those three. So therefore, albumin is not going to be expressed and made in your cells of your eye. But what is going to be expressed is the crystalline because, again, you have the appropriate combination. So this one was orange, purple, pink. So we're going in this direction, orange, purple, pink. So purple's in the middle. And pink. Whoops, orange, sorry. And so you have all of those, orange, purple, pink, all of them combined, and therefore the gene is turned on. RNA polymerase can bond, and you can make your RNA and then your protein. So crystalline gene is turned on in the eye, the lens cell, because of the appropriate activators. So therefore, that's how one way how cells turn on and off different genes is the presence or absence of the correct activator. So we can say albumin here is not expressed just like we did here. No appropriate activators. So down here, let's just go through the liver cell. I'm just going to write a couple of things. Activators required for albumin expression are only in liver cells. And here activators required for crystalline expression are only in lens cells. So that's the synopsis of what's going on here, and that's how different genes, one of the ways that different genes can be turned on in various cells in which they need them. So therefore, this last bullet, and then we'll stop here, the combination of transcription factors binding to the regulatory region at any one time determines how much, if any, of the gene product will be, and you should have produced. Okay? And so this is where we are going to stop, is the control of transcription.